you. Thanks a lot, Cindy. And thanks everybody for being here together. I imagine soon you'll be able to meet in person. I'm going to do share screen here. And then I'm going to start the slideshow. Oh, I can't see the bottom of my thing here, so I have to do it a different way. Here we are. Okay. So is everybody good? Yes, everybody can see. Okay.
Looks great.
Okay, great. Okay. So yeah, so well, this is a big topic, but I'm going to try and show you a few things that would be of interest because of the fact that these fossils that I'll be talking about occur here in Texas, but then put them into the larger context of where we are in time and what we can see here in Texas relating to the whole story of the rise to dominance of flowering plants. And so there's a classic, beautiful picture right there. So we have these, I'm trying to figure out how I can get stuff out of my way here. Here we go. Okay, I'm good now. All right, so so what are flowering plants? Let's first differentiate them from some other things. It's just a simple chart that I found at this very helpful blog site. And here's a list of attributes, so support tissues, true leaves, stems, roots, and so on. And down at the bottom, how do I see, I'm sorry, how do I get rid of the menu at the bottom of my screen? Ah, I figured it out. Okay, so down at the bottom, you see fruits and flowers, and then the last one is water required for fertilization. So of course, that's very essential. I mean, reproduction is the basic, is a basic function of anything that's living. And so what we see is that mosses, they don't have any support tissue. There's pretty much almost everything is absent except for water is required for fertilization. So they're not going to be very prolific or abundant all over the Earth's surface because they can't always be in water and they can't get very big. But when we go to ferns, ferns are a little bit more advanced. So they have all of these things, but they also require water for fertilization. When we get to gymnosperms, gymnosperms begin to have more things than the others, the more primitive kinds of plants. Gymnosperms have pollen, and they also have seeds that occur in a cone. They don't have fruits and flowers, and they don't require water for fertilization, although gymnosperms tend to have what's called a pollination draw. So they produce a liquid that helps the sperm find the ovary for reproduction. And although they have pollen, and the pollen goes through the air very well, they're usually wind pollinated. So they need wind to just blow the pollen on and hope for the best that they land somewhere else where the sperm inside the pollen can then find the ovule and fertilize, and reproduction can take place. Now you notice here they don't have fruits and flowers. Now fruits and the kinds of seeds that flowering plants have, the angiosperms, they are very protective. That is, they keep the seeds, the embryo, insulated from the exterior. So they keep them from drying out. Sometimes sometimes the embryo is so well sealed inside the seed that the plant species will need those seeds to go downstream and be tossed around in the rocks so that the outside of the seed is actually corroded a little bit or eroded a little bit so that they can actually grow a new plant. The pollen of flowering plants is also interesting. the pollen usually has a fairly thick wall, and it has these openings in it, little slits or little pores. And if the pollen begins to dry out as it goes from one flower to another, the pollen will shrink down a little bit and it'll close off the slits in the pores so that they can last longer and take the cell contents of the pollen grain, which has the sperm in it, to the other flowers. So there are all of these adaptations that allow flowering plants to reproduce and to even go dormant. Their seeds can even go dormant for years, many years. We were just talking about how I think the record is 2,000 years. So they have all these great adaptations and then can grow just about anywhere on Earth, even in Antarctica at the edge of the ice. And wherever the ice is retreating, even a little bit, you begin to get the growth of flowering plants. So angiosperms are flowering plants. They reproduce sexually through pollination taking place in the flowers. The seeds are contained within this really great protective barrier called a fruit. And so they're very diverse. There are something like 250,000, perhaps more, flowering plant species. So if we look at the history of plant life, actually we're looking right now at all genera. We see biodiversity during the whole Phanerozoic here. We're looking at, if you pay attention to the green is the well-resolved genera. Those are the ones that we're sure are actually genera. You can see that there are times where there's an increase in diversity, a little bit of a dip here, and then we get more and more and greater diversity as we come towards the present. The earliest angiosperms are represented by their pollen, and those date back to about 130 million years ago. So that's right about here. And what we see here is this K stands for the Cretaceous, and that's because geologists apparently cannot spell. So we have the K here. The beginning of the Cretaceous is 145 million years ago, and the end of the Cretaceous is 66 million years ago. And we are going to focus in on this time period because we go from a time on Earth before the flowering plants to a time when flowering plants become dominant on Earth. And I'll just say as an aside here, because I can't help it, you see that there are these 5 big mass extinctions that are pointed out by these yellow triangles. So here are the 5 big mass extinctions. Mass extinctions mean that there are times in Earth history where over a short period of time, you have the loss of many, many species. And you see that the last great mass extinction occurred right at the boundary between the Cretaceous and the following period. And I will mention that again later. But most scientists now agree, there's a consensus, that we are currently experiencing the 6th mass extinction. So now if we look again here at time and we look at the plant record, we see plant evolution showing the approximate time of emergence of the angiosperm. So that's shown in the light blue. And then everybody else is shown in the darker blue color. Here again is the boundary, the lower boundary of the Cretaceous here, which is in the Mesozoic. That's at 145 million. 130 again is our earliest evidence of angiosperms. And then what we have around here in our neck of the woods right here in Dallas is the Woodbine Formation. And the Woodbine Formation is really interesting because it has plant fossils in it and it also has the fossils of animals in it. So we're going to have a look at that as well as a few other things. If we zoom in now a little bit into the Cretaceous, we again see earliest angiosperm pollen here. Before we get to the Dallas fossils, we have another locality not far away near Glen Rose, and that's called Jones Ranch, and that's in the Twin Mountains Formation. So that's around 112. I might have that wrong. It could be 113. Let's give or take a million years. And then after that we'll talk about Dallas. So what's going on at this early time? That's right in the middle of the Cretaceous. We know that flowering plants were on Earth, right? Because we have the earliest record 130 million years. But what was it like? Was it dominated by flowering plants? As we talked about earlier, what would you see when you walked out the door? So here's Texas, nice and big as it should be. Here is Jones Ranch, located in that part of Texas. And some of you, maybe lots of you have been to Glen Rose. Glen Rose is kind of famous for its dinosaur footprints. And here's a picture, an old picture from the old days, from the work projects administration, WPA days, where they were excavating, and they excavated these footprints here. And then this little guy is sitting in water left over from rain and probably river, and he is sitting in the footprint of a big sauropod, a long-necked dinosaur. And you see that this picture is autographed by Roland T. Bird, who was excavating the footprints. It's pretty cool. So you can see these footprints. If the water level is low, down at Glen Rose, you can actually go down there and see dinosaur footprints. Jones Ranch is very close to Glen Rose, and it's a privately owned ranch, but the ranch owner found some wonderful dinosaur fossils and called paleontologists in to the rescue, and that was an ongoing project for many years that involved lots of volunteers. It involved scientists from SMU, including my husband, Louis Jacobs, and it also involved people from the Fort Worth Museum of Science and History, and it was a great program. It also included school teachers so that they could learn about how to excavate fossils and then understand what happens to the fossils when they go back to the laboratory, and then impart that knowledge to their students. So it was a great enterprise for a number of years. So these volunteers who worked there also found plant fossils. So here's a little fossil map, let's say, of specimens at Jones Ranch. So here's your scale. This is 1 square is 2 meters across. These are all bones, but we don't really care that much. I know I'm mentioning dinosaurs a lot because people like them, but what we care about are plant fossils. That's the really good stuff. So here's fossil wood. There were huge pieces of fossil wood there. And along with the fossil wood, people found lots of plant fossils. And it was really amazing because the rock that contained the dinosaur bones and the plant fossils, the rock, it was really, really hard rock. This was very, very consolidated. And the surprising thing is that these plant fossils were not replaced by silica like we were talking about before. This is actually organically preserved, somehow locked in to this really solid rock. And what these are specimens all from the same kind of plant, the same species. These are the stems, and I'm sure you all know mountain cedar, right? Juniper. So this is not juniper, but it's similar to juniper in that the leaves are very, very teenites. They're very reduced, like you would have on a juniper stem. These little triangular projections are the leaves, and these stems then would have been green and photosynthesizing. And so here's a little bit of a bigger picture of the stem tissue. And they had joints. I see these little crosses here. So these are nodes where you would have the leaves occurring. And if you put some of this organic material from the leaves under a microscope, you would see these openings, which are the stomata. So here are the stomatal openings. Here's one, here's another one. And you can see the cells around it. Those would be just the surface epidermal cells on the leaves. And you see that around the stomatal opening, the cells here are very, very thick. You can actually see how thick they are. And in addition to that, there are these bumps that are protruding into the opening, the stomatal opening. So notice the scale here. This is 2 millimeters, right? So that's 0.2 of a centimeter. This is 20 microns. So this is really, you could not see this with the naked eye. You have to use a microscope to see this. And again, this is all organically preserved. And what this is, it's made up of something called, it's cuticle, and it's made up of cutin. And cutin is another kind of organic material that does not break down as easily as living plant tissue breaks down. So this is a coating, it's actually a waxy coating that leaves have on them to help diminish water evaporation out through the leaf. And it turns out that that was very appropriate for this time period because it was very seasonally dry at this time. So it was hot, it was dry, and these were good adaptations for these plants. And these kinds of plants that are in a family called Chirolepidiaceae, these kinds of plants were very common in this part of the Cretaceous around the world. And this plant is called Phrenolopsis ramosissima. And until we found this, and by we, I mean the royal we, since I didn't actually find it, it was found by the volunteers. This plant was known only, before then, only known from the Potomac group on the east coast of the US. So that was kind of fun. Now, not only did they find the leaves, the shoots of these things, but also male cones. So there are male cones and there are female cones when you have conifers. The male cones are quite small, and here's a 3-dimensionally preserved male cone. You can see all the scales going around here. And here's one in cross-section. And then inside the male cones, of course, they're male, so what do they produce? They produce the pollen, and the pollen has the sperm. And inside that cone, we could dissolve the rock material, get in between the organic matter, and pull out the pollen grains that were preserved in there. So this pollen, it confirms that this is from this plant family because it produces this unique kind of pollen with this ring around the middle. That was a lot of fun. This is looking at the pollen with a light microscope. That is where you shine the light through the slide. So it looks like this with light microscopy. And this is looking at the pollen using a scanning electron microscope. And so what you're doing is you're looking down on the specimen and you're seeing this image because of the way that electrons are hitting the pollen grains. That's the way the electron microscope does it. Here you see the female cones. So here's a big cone, female cone. And inside of these scales, these cone scales, would have been the seeds, just like you would find in a pine cone today, or a juniper cone for that matter. That's where the seeds go. So here's a cross-section, another cross-section, and here's some material from within the female cone part. We didn't find an actual seed intact. And then here's the wood. So we have the conducting cells here. They're not called vessels actually, they're called tracheids in conifers. And here's the specimen out in the field. So you see somebody standing there and you can see how big these trees were. And it was thought before this, that these kinds of plants, in fact, this species was kind of a small thing, but actually it was quite large. So that was a lot of fun. So this site has these fossil plants, and because we have the cones, we have the shoots, the Perot Museum of Nature and Science created a reconstruction of the tree of one of these very plants. So there's the reconstructed tree that's on display with the dinosaurs at the Pearl Museum. Here's the reconstruction of a shoot with little shoots and little male cones still attached. And then these very specimens from the site, the ones that I just showed you, are on display. Yeah. Excuse me. Okay. All right. So, and right next to that is a dinosaur called Tanakasaurus dawsi, and that is approximately the same age as these Jones Ranch specimens. And this though, the Tornadosaurus occurs to the west of Fort Worth. It's about the same age, but it's in a different location. And it's from the area of Weatherford on somebody else's ranch, actually. And at this site, we don't have seeds, we don't have cones and shoots, but we have lots and lots of pieces of the plant cuticle. And again, because the Chirolepidiaceae produce such thick cuticles, sometimes you just get massive amounts of the cuticle itself preserved. So here is a view of cuticle from the Doss Ranch site where you find the Tanakasaurus. And again, we have the same sort of plant. We have these are the stomatal openings, and you get the little papillae, little bumps are overarching. That helps to protect the plant from water loss through the stomatal openings. Okay, so that is one of the sites from our North Texas area. Now let's go to the Woodbine Formation. So now we were at 112 million years ago. And you notice that this locality, Jones Ranch, that I was just talking about, has all kinds of Chirolepidiaceae, all kinds of conifer remains of this species preserved in it, along with the dinosaur, which was called Paleobotanist, by the way. And yet, you don't have much in the way of flowering plants. And it's not just because we're only looking at the one site. This is the way it happens in most of the world, not all of the world, but most of the world. So at this time, flowering plants were just really taking off. They were just getting going. They were very small, and they're usually only found in very careful study of very organic rich kinds of sediments. But again, let's look back at what we have here. Let's go to the Dallas area. Now we're going to go to deposits that are 96 million years old, and that's the Woodbine Formation right around here. So I had a student named Marley Cruz, and she did her master's project on the Woodbine Formation. And she is here collecting pollen samples, pollen and spore samples. And the way you do that is you collect a fine-grained, hopefully organic rich hand sample, and you take it back to the laboratory, which she is shown here on the right, and you have to use a lot of harsh chemicals to separate the little bits of organic matter that will include your pollen and spores from the rest of the sediment. And that's what Marley is doing in this view here. So she went out to 3 different sections. Here's one of the sections that she measured. This is from a place called the ACME Brick Quarry, which I never knew there really was an ACME Brick Quarry, but apparently this is where Roadrunner got all his material. So here's Marley. She is standing there with a Jacob staff, it's called, not having nothing to do with me. And that helps to measure up the section and note down all the different attributes of the sediments and the rocks that you find. So she did that and at the same time collected her pollen samples. Now, back 96 million years ago, there was a shallow interior seaway that went all the way from the old Gulf of Mexico, all the way from there up to the Arctic. And it basically divided North America into an eastern part and a western part. And this arrow shows us, so you are here, 96 million years ago, and the deposition location for the Woodbine Formation. And consequently, the Woodbine Formation, the rocks now crop out. You can find them in a band that's located here across these counties. So here we are in Dallas County, and they crop out, a little bit in Dallas County, but mainly in this region here. So Marley sampled in these three places: Bear Creek, locality 360, and the ACME Brick Quarry, which I just showed you. And she found lots and lots of spores and pollen. And this is what they look like. Marley took all these pictures. These are what are called Trilead spores. They have three seams kind of here. And that is the remains of how they were when they were produced, because they were produced in a tetrad. And they all have different kinds of ornamentation. So they have different names based upon their ornamentation. These are produced mainly by ferns. On the right side here, we have angiosperm pollen, not spores, but pollen, right? Because angiosperms reproduce a different way. And there's a fair diversity of angiosperm pollen. And that's pretty cool. So this is the difference here in Texas between the Glen Rose area and 113 million years ago, and the Dallas area 96 million years ago. So already we are seeing that on the landscape there were enough angiosperms present to produce enough pollen to be found in the sediments. Now there was a master's thesis done many, many years ago where the student looked at pollen from the Glen Rose Formation and found a little bit of angiosperm pollen. So clearly there must have been some around, but not enough to show up in that at that dinosaur locality. So Marley compiled her data. What she did was she counted all the pollen and all the spores on many different slides, many different samples that she extracted in the lab. And this is how we can look at them. This is how we get the information about what the landscape must have looked like in the past. So here you see on the left, we have these pie charts, and on the right, we have a different kind of diagram called a pollen diagram. So if we just look at the pie charts to begin with, this is one of the localities. This is locality 360. And she divided locality 360, which you can look at from this part to this part in this part of the section, let's say. So this is we called a stratigraphic section. To go back. This is a stratigraphic section. So you can imagine Marley is measuring all these rocks going from the bottom to the top, collecting her samples. It's organic rich. And in these samples, she's finding these angiosperms, which in the pie chart go from here all the way around to here, okay? So almost a half of the pie chart in her section, one of her two sections, it consists of angiosperm pollen. The rest of it is ginkgos and cycads, other kinds of gymnosperms. Sorry, it keeps wanting to, I'm pushing the arrow too strongly. Here is the other pie chart, and it shows lots of angiosperms. Here we go from palms, which again are angiosperms. So palms, all of these, all of these, all of these. So we're talking about 2/3 of the pollen grains in this measured section among all her samples are angiosperms. So if we look at the section laid out this way, this puts all the different pollen types in columns, and it shows them in their relative percentages per horizontal sample. And so here, if we just look at tricolpates, let's just focus on that. That's the kind of pollen that angiosperms produce. That's one of the kinds. And you can see how there's a big change in the quantity of tricolpates as you go from the bottom to the top. And of course, the bottom was deposited first. That's the older part of the section, and the top is the younger part of the section. So you do see variation in the amounts and the kinds of pollen that you get through the section. And this is just locality 360. She was asking, well, not only how dominant are angiosperms on the landscape, and can we say that from looking at pollen and spores, but what kind of variation was there from place to place? Now, it's possible or probable that the 3 localities that she looked at come from somewhat different time intervals within the Woodbine Formation. But we can't say exactly how much time difference there is from one locality to another. What we can say is certainly they're representing different kinds of environments. So in the ACME brick quarry, for example, she's divided that big section into 2 different sequences. So one is called ACME brick 1, one is called ACME brick 2, and we see here the angiosperms are shown from this point in the pie chart going around to this point. So what is that? About 60% are flowering plants. ACME brick 2, again, has lots of flowering plants, but we have a lot of unidentifiable kinds of pollen grains. So those may be because the preservation is not very good. So we're kind of missing part of the picture. Here is Bear Creek, the 3rd locality. And again, one place is a little bit different from the other, different amounts of ferns. But here the angiosperm pollen goes from about here to here. So not quite even half. So what does that tell us? That tells us that there is variation on the landscape. Angiosperms have a big role to play by this time. And this is not only the case for the sites that Marley sampled, but it's also the case around the world. So this brings us to the bigger picture. So now we want to expand our understanding of where we are in flowering plant evolution and put it in the larger context of what's being found around the world. And I wanted to just give you one or 2 examples because there's a lot out there. But this is from a study that was done by, that should say Manchester, there's an R there, by Stephen Manchester and his colleagues, looking at one particular kind of plant that came from a site equivalent in age to the woodbine that we find around here. And that's called the Dakota sandstone. And that is also right next to that big inland sea that we looked at earlier. So we're sampling the inland sea right near Dallas, and the Dakota sandstone is up north of us, sampling the margin of the inland sea. And at that locality, they have more in the way of big macrofossils, you would call it, things that you can see with the naked eye, including fossil flowers. So here's an example of a fossil flower. The flower fossils are shown on the left. I hope you can see my pointer. I'm not sure if you can, but here's flower on the left. And we see pictures, various pictures of them in different states of preservation. And in the upper right on the plate shown on the left side, you see some notations with blue arrows, and that's pointing to individual anthers that would have contained the pollen grains. And on the plate that's shown in black and white on the right side, you see the anthers themselves preserved. So picture numbers 4, 5, 6, 7, those are all individual anthers that have pollen grains in them. And picture 9, 10, 11, and 12, those are actually showing you the pollen grains themselves. So it's just amazing that you can get flowers preserved where they grew, and then you can go into the anthers and get the pollen out. This is a reconstruction in the middle here of what the flower would have looked like. So it shows you where the anthers occur right there. And this was then named Dicotanthus cordiformis. So it gets its own name because there's no such thing living today. So what else was happening during Woodbine and Dakota Formation time? We have this, which is the skull of something called Protohadros. So it's a duck-billed dinosaur that was worked on by a former student of my husband's named Jason Head. And this is on display at the Perot Museum as well. This is not the actual original specimen, but it is a 3D print of the specimen that was made after data were collected using X-ray technology for the specimen, for the skull, and then you take the X-rays, just kind of like a CT scan, like you would get in a medical situation, but using stronger X-rays. You take the CT scan, and then the software, you can manipulate that with the software and piece it back together. So the specimen was too difficult to put on display, so they did a 3D printout of the skull. Also known from the Woodbine Formation, actually, is a primitive crocodilian. So this is actually another former ACME student worked on that, Thomas Adams. Yeah, so you had crocodiles, you had an ocean nearby, you had plesiosaurs, you had flowering plants, and so it begins to give you the idea that we had a pretty rich environment around here at that time. So now we're going to move on up to what happened before and then after this big extinction event. So I put a circle around this to remind us that although flowering plants were really taking off in terms of their evolution, and we were beginning to get a high diversity, and we were beginning to see a situation where fossil localities can even be dominated by flowering plants as you go into the late Cretaceous. Well, what happened when you had this event here at the end of the Cretaceous and you had this meteor or asteroid hit the Earth and cause a huge extinction event? Well, I wanted to tell you about this really exciting new report, a new publication that just came out in the journal Science by Carvalho et al., where they looked at many, many localities from tropical Colombia in South America. Okay, so here we are. Here's the general map right here. And here are localities. Some of these localities have plant macrofossils, which you see on the right side, and many, many of the localities are pollen and spore localities. So they have put those together. So now from the same area, they put together all the information from pollen and spores together with the macrofossils. And they have localities that are from just before the impact happened and just after the impact happened. The impact that I'm talking about is the bolide or asteroid impact that has been, most people think, was really at fault for this big extinction event. And the impact crater is shown here. It's about 1,200 miles away from these fossil sites. Okay. So what they found was quite interesting. They show that, well, first of all, let me explain this diagram. This is a change in the proportions of three broad taxonomic categories of polynomorphs, that would be pollen and spores, summarized in 1 million year bins. And these are the angiosperms. I'm going to move mine. So here are the flowering plants. The middle column is gymnosperms, and the left-hand column is pteridophytes, that's ferns. This dotted line here marks 66 million years ago. That's the time of the impact. That's the time of the big extinction event. And when you look at the right side and you look at angiosperms, you see that they were coming in at about 50%. When you get to the impact, you see that they come in at a higher percentage of the paleofloras, but it's not until about 60 million years ago where they get really dominant. If you look at the gymnosperms, they take a huge hit, and that happens in almost all places around the world. Of course, we still have gymnosperms. We have big boreal forests up in Canada, and we have them down south. But the diversity of gymnosperms goes down by a lot, in the tropics especially, and at middle latitudes. And also, we're pretty close to the impact crater. The ferns do better. Ferns tend to come back pretty well, and it's not until the angiosperms really take off that they push out the ferns. So this is really a beautiful summary of what happens towards the end of the Cretaceous and then going into the Cenozoic, which is where you have then the rise of mammals, and you reach this eventually you reach this modern world that we live in today. I wanted to mention to you before I finish that some other kinds of fossils that people look at to understand the origins and the diversification of angiosperms are charcoalified flowers, charcoalified bits. And so in the very early days, so equivalent to that Jones Ranch locality where I showed you the Chirolithidiaceae, and a little bit younger than that, people have gone to these very organic rich deposits in Portugal, for example, and separated out the bits of charcoal from the rest of the sediment, and then under a regular dissecting microscope, picked out individual flowers and other parts of these early flowering plants from the matrix. So these all were very tiny. These flowers are only on the order of a couple of millimeters in size. So 1/2 to 2 millimeters long from Sweden and Portugal. That's what this summary picture is. So this is scanning electron microscopy of complete flowers. And this is a great summary, I thought, of the whole story altogether. It's a nice visualization of the rise to dominance of flowering plants. So here we have the Cretaceous, we have the early and the late Cretaceous here. We go from the Albian and we then pass into the Cenomanian. This time right in here is where we have the Jones Ranch locality. So you have a lot of these conifer type things, these conifers, you have ferns, you have cycads and things that look like or related distantly to cycads. And the flowering plants that you have are going to be very small. But when you get into the Cenomanian, you start to get a greater diversity of the flowering plants, and they begin to get larger. And by the time you get to the very late Cretaceous, you have much bigger flowering plants, and you increase their diversity by a lot. When you get to the early part of the Cenozoic, especially the Eocene, so this is not the very earliest, but this would be about 6 million years after the very earliest, not 6 million actually, about 15 million, you get to the Eocene and your diversity really goes way high. And you begin to get a world that's much more similar to the world that we live in now. You get birds, you get the coevolution of, well, actually even down in the Cretaceous, you get the coevolution of insect pollinators that go along with the angiosperms. And so this is just a summary of how angiosperms really take over. And that is all I have. So I'm going to stop sharing.
Thank you so much.
You're very welcome.
We don't have any questions yet, so please, everyone type away or since we don't have any, just unmute yourself and speak up.
Well, round of applause for a really fascinating program.
Oh, thank you. Good.
Yay!
Why do you think the angiosperms proliferated so before and after the impact?
Mm-hmm. The angiosperms have this whole suite of characters that makes them really good at staying protected in a dry climate, in a wet climate, in a cold climate, in a hot climate. They have this amazing proliferation of adaptations so that they reproduce. In fact, their whole reproduction takes place in a very small space, a very small and protected space within the flower. So you have the pollen grains are transporting the sperm, and the pollen grains themselves are pretty hardy. They can close down and protect the plants or the pollen contents from the loss of moisture by closing down. And a lot of flowering plants, in fact, the more primitive flowering plants, used insects and other animal pollinators to deliver the pollen from one flower to another, or from one flower on the same plant to another. So they were very directed, and that also helps to make pollination happen despite the fact that they're not in water like ferns have to be. So that's part of the series of adaptations. The other thing is that they produce these seeds that are extremely well protected. So the seeds then can get dispersed and leave the plant and go and germinate where conditions are right, but they don't have to be transported by water or be protected from the exterior environment. And the same thing goes for the leaves. And leaves develop these means of protection. But you have all of these things together in flowering plants that you don't have all together in gymnosperms and more primitive plants. And so that makes them really robust, and they also have some chemical adaptations, many actually chemical adaptations that make them able to resist insect herbivores. They have chemical adaptations that help them to photosynthesize efficiently in dry climates. And when you add that to everything else, they just can do really well. They live everywhere.
Did flowering plants, did angiosperms emerge from exosperms, or did they have a common ancestor further back?
Well, they would have, yeah, so there's still some discussion about their ancestry, which is interesting. It's a little bit of a challenging mystery. So, but yes, they would have emerged from some kind of gymnosperm ancestor, yes. So if we look at that, remember that chart I showed with the blue, the pale blue is the angiosperm, and then there were some potential prospective possible ancestral groups, but they weren't shown attached because it's not certain.
We have some other questions coming in through chat.
Okay.
Someone would like to know if we could post a list of some of the places discussed so that they could visit. Is there any of those places they could visit besides Glen Rose? Like, I guess I'm assuming the ACME Quarry is closed to public.
Yeah, unfortunately. Yeah, the ACME Quarry is closed. Locality 360 is right along Highway 360. So that place could be visited because that's public land. The location, you know what? I would advise that anybody who wants to get to fossil sites, that they go to the Dallas Paleontological Society website and start chatting with people there, because they know all of the accessible places to get to that are available to the public. Locality 360 though is, I could tell you how to get there. There's a trestle, like an old train trestle that goes across the highway. And, you know, you have to pull over onto the side of the road and right before that train trestle. And there used to be a really nice exposed cliffside by the road. It's a little bit grown over, but you can walk back into there's a dirt road that goes back in there. You can see outcrops, and you start digging around the black layers, and you'll find plant fossils. And there's pollen in there too if you're able to look at it.
And which society is that again that we should?
It's called the Dallas Paleontological Society. There's a very active group of well over 100 people who regularly, they have a meeting every month. And so if you want to join another group, that's a good one to join. They're really good.
Speaking of that, a personal question, and that Glen Rose photo, was that Chuck Bensley in the middle?
I think it was Jim Diffley.
Okay.
Yeah, I'm pretty sure it was Jim Diffley from the Fort Worth. Well, he's retired now, but at that time he was at the Fort Worth Museum of Science and History.
Most of us know Rosa, so.
Yes, of course. And yeah, they were very much involved with the Dallas Paleontological Society as well. And of course, Chuck goes way back with the museum when it used to be the Dallas Museum.
I've lost touch with Rosa though. Is she still around? And
I believe so.
Yeah.
I have a question for Bonnie.
Sure.
When that meteor hit 66 million years ago, do you think it had anything to do with herbivore predators on angiosperms, that those predators were wiped out and that allowed angiosperms to proliferate and become more dominant as time went by? Do you think it had anything to do with these herbivores that were wiped out?
I imagine it could have. People do talk about that. I mean, that's one of the ideas. I'm going to ask if my husband can hear, because can you hear it? Oh my goodness. Okay, you can hear me now, right?
Yes.
Okay, now we both can hear you. I didn't have sitting here in silence the whole time. Okay, so yes, the question was whether the loss of the big herbivores could have had something to do with the rise of angiosperms after the impact. And people have said that, but I just don't know. That may have been true for some groups. Those big herbivores, weren't, I'm sure they were eating some angiosperms, yeah. But they were also eating a lot of other things. They ate things that you wouldn't think anybody would eat, like they ate cycads, which are, we all know are poisonous to our pets and everything. So they ate cycads, we know that. They ate other kinds of conifers, gymnosperms. And the only reason we know that is because Karen Chin is a paleobotanist who looks at the remains of plants inside dinosaur coprolites. So, Louis, do you have something to add to that? No, he doesn't have anything to add.
Thank you.
Sure.
Lana would like to know if were there any other environmental factors that contributed to the rise of dominant flowering plants other than more time for them to develop and adapt?
I would say that, everything evolves in its time. It's kind of that sounds kind of mushy, but you can't get to flowering plants until you get to their ancestors. And once this sweep of characters, these innovations, let's call them, evolved, it allowed the proliferation of flowering plants. So it's not that they had more time than other things. They came along later, but we still have gymnosperms, we still have them around. and yet they're not nearly as diverse. In fact, they probably likely were outcompeted by flowering plants.
Were they able to take advantage
So,
of those territories?
yes, flowering plants were able to take it. Thank you. I got a little key over here, little nudge. Yes, that flowering plants were able to take advantage of disturbed areas. They could get in there. They have part of what they can do is grow fast. They can produce many seeds. Some of the earliest of the flowering plants could produce many seeds. They were small. They might have grown near water. They may have, taken advantage of places that perhaps gymnosperms couldn't. But yeah, does that answer the question?
I hope so. Ernie said, I think you, oh, sorry, Lana, did it? You're muted. You can nod. Oh,
I think so. And the previous person's question about herbivores, and their effect, it was also really interesting. And I think kind of tied in with my question as well. But it sounds like just all of their great adaptations is really what allowed them to really take off more so than environmental changes. So thank you.
You're welcome. Yeah, I think that's true because, actually in looking at diversification, it does seem that when the Earth was really warm, when you get to the Eocene, so the Eocene started 55 million years ago. Oops, I'm frozen. Can you hear me?
yes.
Okay, good. So when the Eocene comes around, you get really hot climates. You have deciduous plants all the way up to the polar region, and you have tropical settings going all the way far north to the mid-latitudes. And pollen records and plant records show a lot of diversity during that time. It's not exactly completely clear what exactly was going on all around the equatorial region, but so there may have been times that the environment would have made it more, made it better, generally speaking, for plants anyway. But I think because flowering plants can do all this great stuff, they can make it in almost any environment.
Well, all of this raises the question that whatever pressures, environmental pressures there were that caused the multiple adaptations of flowering plants must surely have been pressing on, say, gymnosperms as well. Why didn't they develop these adaptations? Or am I am I missing the story that they did and those adaptations are called flowering plants?
Well, yeah, that's probably more like it. Yeah, right. I mean, right. I mean, when you get something like one of the adaptations, for example, is double fertilization. So that allows the flowering plants to where fertilization takes place in the ovule, and the seed can grow and the fruit can grow, and inside the seed you have nutritive tissue, you have endosperm. So that's another adaptation that they had. And almost no gymnosperms have that. What's really interesting though is that you find here and there among primitive gymnosperms and among some of the dead gymnosperm groups, the ones that went extinct, you have like little inklings of, going towards what you would think of as an angiosperm. So it turns out that, I don't know if you know the plant ephedra. Ephedra, yeah, that's where, it's a stimulant, that's where ephedrine comes from. So ephedra is from a seedless vascular plant. It's a gymnosperm, it's not an angiosperm, and yet it has double fertilization. And it was really common. The ephedras, and there's a remnant relative of ephedra called Welwitschia that only grows in the Namibian desert and the desert of southern Angola. These things are remnants of what they once were back in the Cretaceous, and that thing developed double fertilization. It's the only one. And then there's another relative of ephedra called Needham, which is also very restricted in its range. Ephedra actually grows in a lot of places. The other relatives don't. They're just little remnants. And that Needham makes a leaf that looks a lot like a regular dicot flowering plant leaf. Whereas all the other gymnosperms, that you can tell, like we can tell what a conifer leaf, they look like needles, and they look like even Podocarpus, which has, it still has a very linear kind of a leaf. Yeah, so it's funny how that, maybe those environmental pressures were pressing on those. I hate to think of it that way, because mutations, they happen. Things that work, they stick around, and things that don't, they don't.
So I guess the answer to my question is yes, the gymnosperms did evolve those adaptations, and we call those adapted plants angiosperms.
I guess so. The angiosperms successfully put them all together.
Yeah.
And then allowed them to really take off.
Ernie would like to know, he says, I think you said the pie graphs were not dated and may have come from different periods. Why couldn't you date those using radionuclide dating methods, or did he misunderstand?
No, that's a good question. So, there is one date related to the Woodbine Formation that got processed recently, and that date is 90. Right, exactly. 95 million, but it's not from within the Woodbine Formation. And the reason is because not all kinds of rocks and sediments have the right kinds of elements in them that allow for radiometric dating. So sometimes you just don't have the benefit of those kinds of rocks. If you have volcanic rocks, they have crystals that start the clock, basically. They have radioactive elements in them. The clock starts ticking, they start decaying, and you can use those for dating. The Woodbine mainly has been dated by some of the marine fossils that then are correlated with marine sediments that have dates on them. So, but the closest, you can only get within a certain range, and we know it's about 96 million years old. But the other way of getting at it is, let's say it's 96 million years old here, and you have sediments that are right below it. Well, these are older. You may not know how much older, but they're older. The problem with the Woodbine is that they occur at isolated outcrops that you can't necessarily trace them right back together again. So it's probably not a lot of time represented between those three sites, but it's not as, you can't precisely say it like you could if you had radiometric dates.
That was a good question.
Yeah.
Barbara would like to know, I found fossils on my property in Graham, Texas. How can I find out if they're plants? If they are plants, yes.
Okay. Well, there is a book called Common Fossils of Texas. I believe that's the title. And the common ones will be in there because they do include plant fossils in that book. What county is Graham in?
Young County.
Where is that?
Which county?
Young?
Young County.
Where is Young County?
Yeah, what town is it?
Northwest of Fort Worth
Oh, north.
by several counties.
Oh, okay. So maybe not too far from Seymour?
We're next to Palo Pinto is one. Palo Pinto County.
Oh, she froze.
PK Lake.
Oh, Bonnie froze, not you, Barbara.
Yeah,
Yeah, so Palo Pinto County has rocks of Pennsylvanian age, so about 270 million years old, and they do produce plant fossils, and those plant fossils are going to look like the leaves look like little ferns, and then there's some other ones. So if you look up plant fossils of Palo Pinto County or fossils of Pennsylvanian age, that'll probably get you to get a kind of visual search image for what you might find. I think that's probably the best way to do it if you Google that.
Okay, thank you.
You're welcome. There's been a lot of work done from the Smithsonian and others up in the whole Wichita Falls, Seymour area, all the way up there. And I went collecting fossils once in by Gordon, Texas, which is in Palo Pinto County.
Okay.
And we found, it was somebody's ranch, but we found a lot of fossils.
I thought maybe it was like a crocodile tail, because it has ridges, and it's like a solid log with ridges. But somebody said it was a plant.
Can we see it?
Okay.
I will make you a co-host. While she's getting that, we have one last question in chat. Another Barbara wanted to know, were angiosperms first only wind pollinated, and did insects co-evolve with the angiosperms?
Yes, that is such a great question. Well, it turns out that it seems that probably they were insect pollinated first, and then they were wind pollinated. And I was going to include this in the talk, but it was already getting too long. So I didn't. I want to go back to my screen in 1 minute. I want to see if I have it stuck in at the end. I don't think I do though. Let me try.
While you're getting that, Barbara, you're very dark. Do you have any more light?
Can I send some follow-up information to you? There was this paper that has the most gorgeous diagram in it that shows the evolutionary history of all the flowering plants in like a chart. The middle would be the primitive, and out towards the ends are the more advanced. And it puts all the groups into whether they're insect. Okay, hang on a second, Barbara Morrison, 1 minute, and I've just finished answering this question. And I will send this to all of you if I can. I'll send it to Carol or Cindy.
Yep, either one will work.
Okay, great. And now Barbara, sorry, go ahead.
Okay, I don't know if you can see the
Can you look at
picture or
this,
not.
Lois?
Yeah.
Okay.
It has like a ridge down the center, and then it has side ridges as well. It feels like rock, but it's tan in color.
Keep it there for a second.
Yeah, see what it is.
Is there anything? Oh, there.
Oh,
That
there you
help?
go. I see what it is. Yes, I see what it is. Okay, got it. So that's a relative of a horsetail. That's a plant.
A horsetail?
Yeah, it's a relative of a horsetail. It's called Calamites.
Okay. That
yeah.
sounds, somebody looked at it, and I think that's what they said, a horsetail.
All right.
Thank you.
You win a gold star, Bonnie.
Gold star. All right.
Horsetail.
Yay, I can tell calamites. So they had whorls of leaves, like, you know, I don't know if you've ever seen horsetail. Today they're really reduced little things, but calamites back in those days, the Pennsylvania used to be really big. It could get really big, and its relatives could get really big. And so they had these axes, you know, these linear axes with ridges all along them, and then at each node they had
Yeah.
leaves that would come out around the node.
Okay. Is this old?
Yeah, it's old. It's like 270 million years old.
Holy cow.
Yeah.
Don't use it as a doorstop anymore, Barbara.
No, I have it in a special drawer.
Isn't that fun?
I found it in my orchard when I was planting my trees, and I thought this is something that's important, and that's so I've saved all the pieces.
Good.
Okay.
Well, if there's one there, there's more there.
Yeah, there's probably more. Yeah.
Yeah, I had found a number of pieces, but yeah,
I mean, maybe you can find some other kinds too. You know, more of the ferny looking ones.
You might have a forest there, a fossil forest.
possible. Thank you so much. I appreciate your time. Very interesting.
Sure.
I think that's all we have. We don't have any more questions. Anybody else want to? Lana, I see you. Go for it.
Just for fun. I know that all the time, you know, new things are being found that can kind of change, you know, what we thought was. So do you think that, you know, maybe something could be found one of these days that changes the time period of angiosperms to maybe further back? Or do you think that based on the evolution of things that's really very not likely?
That is a wonderful question. And the answer is yeah, sure. It could go farther back. I mean, you know, there's been a lot of searching. But I always hate it when people say, oh, well, you know, if it was there, it would have been found by now, because, you know, it's not necessarily the case. Now, having said that, you know, we're not going to suddenly find flowering plants from 250 million years ago, you know, but we're not going to find it from probably even, you know, 200 million years ago. But you know, maybe yes, back into the latest Jurassic. Yeah, that's very likely. And we just don't have them. We don't have them now. Yeah.
I love the idea that that we don't know everything, and that there's always something more out there to discover that might change the way we, you know, see the world and the history and everything. So cool.
That is absolutely true. There's so much to be discovered. And that paper that I showed you about the Columbia, the reason I was so excited about it is because it is the one and only record in the world of that transition from the latest Cretaceous into the Cenozoic for tropical flowering plants. I mean, and they put together, not only is it the only, but they put together the macrofossils with the pollen. And what they showed actually from their study was not only how the percentages changed, but really interesting was what they showed was that in the late Cretaceous, there was a forest there, yeah, but it wasn't like the modern tropical forest. It was not stratified. So the modern neotropical forest is one of the richest in the world. And the trees are stratified. You know, you've got the emergents that are super tall, and then you've got another layer, and then you've got another stratum, and they're hyper diverse. And that is not the way it was at the end of the Cretaceous. So in a way, the bolide came and kind of knocked things back enough that then you got the emergence of these amazing modern type forests. And they got that way within 6 million years of the mass extinction. So that is totally new information. And that was just published like 3 months ago.
Could matching the pollen with the other macrofossils be likened to kind of like when archaeologists match finds with potsherds to help time them, date them, I mean? Is that kind of a similar?
You could if you had, yeah, so right, what you're getting at is, so if you have pollen from species A, and you have leaves, you might put those two together, or you might say, or is that how you're saying, or are you saying that you can use the pollen sequence as a chronology, a time marker?
Yeah, like, because you mentioned something a few minutes ago and I already forgot what exactly you were saying, but when they found the pollen and macro fossils, I don't know
Right.
if that was of plants or if those were of
Oh,
like animals. So like, do they sometimes use other fossils to help date some of the plant fossils the way sometimes in archaeology, if they find something along with potsherds that they know that this style of pottery was from this era.
yeah,
Is
exactly.
it
Yes.
okay.
Yes. So yes. So in some places where, say where the pollen grains are very well known in a sequence, and oil companies use this all the time. They use what's called biochronology and biostratigraphy. So they tie in tight sequences of changes in species of, say, pollen grain. They don't even know where who belongs to those. They don't know what kind of plants produce them. Maybe they know ferns, but they just give them names that reflect their shape or the number of pores they have or something. And then they get the sequence dated somewhere using, good dating techniques. So you could possibly be in a place where you've got lots of pollen in the sequence, you've got the dating from that, and then you here and there you have your plant macrofossils, and you can line them up in the stratigraphy and then, yes, know how old they are. You could do it like that. And then you can even add to that by saying to yourself, well, maybe we can figure out who's producing these kind of pollen grains. And that's a huge prospect, but yes, it's another goal, is to figure out, what pollen grains that we have that are so abundant, let's say in the late Cretaceous, what plants do they actually go with? And some of those are not yet known, but that was one of the goals of looking at those charcoalized flowers that are so tiny, because then you can just go in and look for the pollen. Yeah, good question. Yeah, good question. Yeah.
Very cool. Well, thank you so much, Bonnie. It's been such a wonderful evening. And I'm sure everybody's agreeing with me in the chat. So thank you very much
I enjoyed it.
For waiting 2 years for us.
Oh my God, waiting 2 years for you. You waited 2 years for me. Thank you so much. It was really enjoyable. I really enjoy it. I'm giving a shout out to Doug Reinelt because I know him from SMU. Hi, Doug. Okay, thank you all so much.
Bye. Have a great evening, everyone.
Bye-bye. You too. Have a great evening. Bye.
Thanks.